comprehensive protocol. So now we're going to turn, hopefully Steve is ready in the next 15 minutes or so. Steve Percival, Dr. Percival from England, is going to give us a comprehensive view of the basic science and really kind of the rationale of the mechanism of action of betaine and PHMB and the way Prontosan works. So, Dr. Percival.
Lovely. Thank you very much indeed. And it's nice to be speaking to you from sunny England. Over the next 20 minutes, I'm going to give you an overview in reference to microorganisms, biofilms, and touch on these irrigating solutions that we're talking about today. But when we think about microbiology, what we always have to bear in mind, we're dealing with bugs that are either swimming around, and these are called planktonic bugs, or we have bugs that attach onto a surface. And when they're attached onto a surface, they're called sessile, and they grow into these things called biofilms, which really has only entered the medical profession, I'd say, in the last 15 years. So these microorganisms actually exist in both states. But of course, without being too anthropomorphic, what we've got to bear in mind is that bugs do prefer a surface because there's a number of reasons ranging from protection to getting a food source and also in terms of interacting with other microbes, because there is a lot of biological warfare that's happening in the human body as well as externally to the body. So there's natural competition going on all the time with different bugs. And of course, when we look at the human body, we know we have different sources of microorganisms that are in the gastrointestinal tract, the urogenital tract, the mouth, the skin. And we know these are moving around all the time and they're very diverse. And of course, what can happen is when you get these bugs in different parts of the human body where they shouldn't be, ultimately they lead to problems. So, for example, in this diagram here, you'll see different areas whereby medical devices have been put into a host and they've got a biofilm related infection. So number 3, for example, is keratitis of the eye, often due to acanthamoeba. And these often grow as biofilms on contact lenses. You'll get cochlear implants, you'll get biofilms on tricuspid and bicuspid valves. A major one on number 7, of course, are intravascular catheters, where the bugs actually grow interluminally inside those catheters. So we develop these catheter lock flush devices that hopefully try and remove biofilms. So anything from a prosthetic perspective has the ability to allow bugs to grow on them and then ultimately over time grow as a biofilm. But if we get a biofilm, that represents a major issue in terms of managing those microorganisms. So when we look at the host and we look at biofilms in general, we have them throughout the human body. So a good example may be the tongue. Another one may be your teeth. And imagine what we do to our teeth all the time to manage that biofilm. So we brush our teeth in the morning. Some people may use the mouth rinses as well to keep the microbial load down. We can get them, as you know, on your urinary or causing urinary tract infections, particularly forming on Foley catheters where they cause encrustation due to a bacteria known as Proteus mirabilis. And of course, that will block up the inside of the catheters and ultimately lead to an infection. We get them in our drinking water, and these are tubercles that you find on cast iron pipes on the right-hand side there. And these are very thick biofilms that you can physically see. Again, on ships' hulls, these form and will increase the drag on ships. Ultimately, that will lead to global warming because those biofilms increase the drag. And for that boat to move faster in the water, it has to use more fuel. So this is a major issue in terms of biofilms. Endotracheal tubes, as you know, ventilator-associated pneumonia is linked to biofilms, particularly on the dead legs there, on endotracheal tubes where you'll get biofilms being formed. And even in your toilets, there's a lot of work taking place on detergents and disinfectants that control biofilms there. So we've got to then establish how biofilms form. and they will form in acute wounds, in chronic wounds. Any incision that's made, if there's microorganisms there, they have the ability to proliferate, contaminate, and colonize a surface. But the idea is we don't want those bugs to actually start multiplying, growing, and causing problems to the host. So when bugs are in the vicinity of a surface, but bear in mind that bugs don't have to attach onto a solid surface to grow as a biofilm, but when they're around, they can attach to each other and start multiplying and growing, and they can then attach onto a surface as well. And once they're on that surface, they start coating themselves in extracellular material, and that aids their protection. And that becomes like a living organism inside the host. But we always have to remember when biofilms are forming, when they bugs attach onto a surface, often they're not attaching to that surface per se. They're attaching onto a thing called a conditioning film. So anything you put in the human body will be conditioned straight away with proteins. So those bugs are actually attaching to that conditioning film, not the surface per se. And when you have a shower tonight, by all means, look at your shower heads and you'll see that there's biofilms inside your shower heads that may contain mycobacterium as well as Legionella. So it's always something to think about as well as when you have your drinking water here in the drinking water pipe. The issues with biofilms are numerous, and I won't go through all this, but in terms of a medical issue at the moment, the major issue you have with biofilms is their tolerance to antimicrobials. So once these bugs start growing on a surface, what happens is they become very recalcitrant towards topical antiseptics as well as systemic antibiotics. And if you've got patients that are very ischemic, of course, those antibiotics can't get to the site by where the biofilm has formed. Now, this is only a tolerance, it is not a resistance. So, what this means is once the bugs are in the biofilm, they can be up to 1,000 times more tolerant to that antimicrobial. But if you take them out of the biofilm, you can kill them quite easily. It's similar to how you'd kill them in the planktonic state. But this tolerance isn't genetic. So it can be due to the structure of the biofilm. It can be due to the fact that bugs within the biofilm become very slow growing or dormant. So any actual antibiotics that require a bug to be multiplying, if these bacteria go dormant, things like penicillin won't work because, of course, they work on their cell wall. So we have to bear this in mind. We don't want a biofilm being formed because it becomes very problematic to kill these bugs. And within 10% of any microbial population, we have these things called persister cells. So anything that's reoccurring in terms of infection, we have this population we can't kill. So when we think about these persister cells, if we apply an antimicrobial to them, what will happen is you'll select for these more tolerant microbes. And if you take away that selective pressure, what will happen is you'll get regrowth. So any form of infection, any chronic condition that keeps reoccurring, you know you have a biofilm, and that becomes very problematic. So you can look at various markers to determine whether you have a biofilm related surgical site infection that you're trying to prevent. And all these potential markers, if you have them, ranging from uh, recalcitrance to antibiotics, ranging from reoccurring inflammation or chronic and localized infection, these are very strong indicators you have got a biofilm. So that becomes a different way to manage that patient because then it becomes guided biofilm based management then in terms of that patient. So when we look at prosthetics and biofilms, we know that a lot of these biofilms, these are the things that are causing or having effect on the infection rates. And infection rates are exceptionally low in terms of the prosthetic joint infections compared to things like chronic wounds, of course, particularly when you have patients with diabetes who get diabetic foot ulcers. So what we have is an awful lot of evidence that biofilms are found on prosthetic devices, of course. So there's a major emphasis to try and control these adhesion of bugs onto these surfaces, as well as controlling biofilms. But even when biopsies and swabs are taken, often these recover no microbes, and they're often culture negative, because what happens is when bugs grow in a biofilm, they change their whole physiology and they take a longer time to grow on agar plates. So when you're looking at antibiotic sensitivity, for example, to determine which bacteria is causing the problem, you'll find that often you'll have to leave that plate incubated for a longer period of time to determine which is the major problem bug here we're trying to manage. So as I'm saying, when it comes to biofilms, when it comes to microorganisms, you want to try and stop them attaching onto the surface. You want to try and stop them forming a biofilm because that causes major problems to the patient and becomes a clinical challenge because there's so many variables you're dealing with. So we have to apply different methodologies. And one, of course, that's employed routinely is irrigation when there's any sorts of prosthetic device going into a patient. These play a critical role in terms of preventing the formation of biofilm. If a biofilm is starting to form, they can help disruption. And of course, it's crucial that these irrigation solutions have some form of antimicrobial effect. Of course, this will help clear the actual bugs from where the site is being operated on. So we look for some ideal sorts of solutions that can be applied in these situations. So we're looking for solutions that can get at least a log 4 reduction. So the FDA have guidelines, of course, when you're developing an antimicrobe, to call it an antimicrobial, you have to achieve a log 4 reduction in terms of that microbiological load. We also have to look at the cytotoxic effects of these antimicrobials. So as we know, when you develop a product, we have to do biocompatibility testing, and there's cytotoxicity studies that have to be undertaken. So it's important that we don't have a major effect on the patient's cells because they're important for the healing process, of course. But we also have to make sure when it comes to antimicrobial stewardship that we are dealing with antimicrobial agents that have a risk both to society, the patient, and also have an effect on what you're selecting for in that patient. And if you don't know the limitations of antimicrobes you're using, that would be classed as inappropriate use because these limitations will affect the therapeutic effect of these antimicrobes, particularly on biofilms. And there's a lot of synergy between antimicrobes as well. So a very important one is organic matter. And what I mean by that, if this everything in terms of an antimicrobial performance is affected by a biological demand. So if there's lots of biology there, lots of cells, you will reduce the efficacy of that antimicrobial. Contact time is exceptionally important. So you have to leave the antimicrobial in contact with the bugs for an appropriate amount of time for it to become effective. So like during COVID, of course, we were using disinfectants all the time. A lot of people weren't leaving them on the surface for long enough to actually kill the bugs. And they were being wiped off very, very quickly. And ultimately what that will achieve is you're not reducing the numbers of bugs on those surfaces. Concentration of the antimicrobe is very important. You want to reach therapeutic levels. But when you have a biofilm, bear in mind when you do an antibiotic sensitivity test, that is not bugs on a biofilm. So when you're using an antibiotic, you have to bear in mind you have to increase the concentration if you have a biofilm-related infection. And as we know, a major thing now, particularly with antimicrobial stewardship and anybody producing an antimicrobial, the requirements by the FDA are you have to now show that that antimicrobial you're using doesn't have a cross-resistant effect on enhancing antibiotic resistant genes. And of course, there's no standards for this, and we have to look at genetics, etc., genomics to understand which genes are being switched on. But it's very important that the use of things like chlorhexidine, for example, which is known to cause a cross resistance, as well as triclosan, as well as benzalkonium chloride. These things, of course, now are not put in like cosmetics because of this issue. So this is very important when you're evaluating which antiseptic to actually use. So the objectives really in the management of any sort of prosthetic joint infection, an irrigating solution is used to reduce the microbial load. The idea is you don't want a biofilm to form, so you're trying to prevent that. And to prevent that, you've got to prevent bugs attaching. You've got to have something that will support the natural healing process of the body and optimize that. And of course, you want to prevent regrowth, hence why you brush your teeth. You're preventing those biofilms from keep reoccurring all the time. So key irrigating solutions that are employed regularly are things like chlorhexidine, your guaninines, your PHMBs, your povidone iodines, as well as saline. And there are a bunch of others that particularly are used in Europe, probably more so than the US. So when we look at the pros and cons of these, they'll have a different array of positives and negatives. So chlorhexidine, for example, there's an array of data out there showing that it's very effective on skin, and it's employed particularly for skin preps as well as iodine. However, we know that chlorhexidine is very cytotoxic, particularly at high concentrations. There are issues with cross resistance, and when it's used as an irrigating solution, we know that has to be washed out or irrigated out with saline to reduce this cytotoxic effect. Similar to that of iodine, of course. Again, iodine's very effective. It's particularly when it's used for dog bites as well. That's the standard protocols of care in the UK. And we know that it can be very good on biofilms. There's, I think, only one incidence of resistance to iodine that occurred in the 70s, so it has a low level of resistance. But again, this one can be affected by organic matter, and again, it can be very cytotoxic. Another one is saline that's used routinely, generally regarded as safe, of course, but unfortunately has no antibiofilm abilities and has no effect on microbes. So then you move on to PHMB, of course, and a lot of these PHMBs are combined with surfactants that are very important. And we know they have a broad spectrum of activity. They're generally very low in cytotoxicity and different to the other ones. This doesn't need to be washed out of the wound, of course. And it seems to be very efficacious even when there's presence of organic matter. So it's been shown to be very effective in terms of killing bugs in the planktonic state as well as within the biofilm. Surfactants are very important when we look at these irrigating solutions now, because surfactants change the surface tension between surfaces. And these surfactants have the ability to prevent microorganisms attaching onto the surface. So you'll find things like Prontosan have a surfactant called betaine, but also don't forget PHMB is a surfactant as well at low concentrations. And there's even evidence to show that it can help with cellular reinvigoration, particularly cells that are undergoing necrosis, not apoptosis, but they've been shown to reinvigorate some cells. So these surface active molecules are very important, of course. And they become more important if you leave them, particularly on hardware, provided they don't cause any corrosion. But what you'll generally find is, as I said, anything that goes in the human body will be conditioned with a film. And that is generally protein, which deposits on that surface very, very quickly. So any micro contouring, any surface changes you've made, these can be counteracted very quickly when they're put in the body. So what you're trying to do, if you can get a surfactant on the surface, what will happen is a bug, when it goes to a surface, twitches a lot and determines whether it will attach and stay there. If you've got a surfactant on there, what will happen? They will actually detach from that surface because it's not favorable for them to grow on. And if they can't attach, they can't form a biofilm, you won't get regrowth, and you're preventing the formation of a biofilm, which is very beneficial as that hardware is in the patient. Now, there's lots of studies that have been taken place on irrigating solutions, and one study looked at irrigating solutions such as Prontosan, which has the PHMB, Iricept, which has the chlorhexidine, Experience, which of course has citric acid, and Sure, which has the povidone iodine. A study was taken place to look at these bugs in more dirty conditions. And what I mean by that, they've got lots of protein and blood in them, and it's following a standard. And the idea is if you can achieve a log reduction of 5 in more dirty, more realistic conditions, these have been shown to be very effective in terms of the human body. So when you study all these irrigating solutions, you find some can be very effective very, very quickly. So Prontosan, for example, within 1 minute is achieving a log reduction of 5, particularly against Staph aureus, compared to the other irrigating solutions. Been the same with Enterococcus, the same with Pseudomonas aeruginosa, and of course they can vary depending on the bugs you're dealing with. And we're continually getting significant antimicrobial effects with Prontosan compared to some of the irrigating solutions. And as you're following a standard procedure here, everything is standardized and you're getting more realistic data here that can be applied to the patient. And Cutibacterium, of course, used to be propionyl bacteria, acnes, again, very effective on that, which is often very hard to kill. We also know if you look then at these different solutions on biofilms, we know we do get a reduced effect on biofilms, which happens with any antimicrobial. But of course, what you're finding against Staph aureus and Pseudomonas aeruginosa, Prontosan is having a significant effect of here. The PHMBs have an effect. And we can study these actually under confocal laser microscopy. So if you have a look on the left, this is a biofilm where all the bugs are living, so they're staying green. We then apply a PHMB solution, and after 24 hours, the bugs and the biofilm are being removed. And once they go red, those bugs are dead. So we can show in real time how quickly we're killing these microbes and killing the bugs within a biofilm on a surface. So that ultimately shows that effect there. And what we've got to bear in mind as well, when we look at biofilms, and if they have actually formed, as you can see here, there's a biofilm here, we really want to get rid of them very, very quickly because we don't want them to carry on growing. However, if you're coming to manage a biofilm, you have to look at the collateral damage that you're causing. So a biofilm is kept together with extracellular polymeric substances, which are polysaccharides, proteins, and metal ions. What happens is if you break up a biofilm, these enter the human body and they're very immunogenic. So you can prolong the inflammatory response here. You release lots of toxins and enzymes. Biofilms are released, metal ions, aggregates of microbes, extracellular DNA. And all these have an effect on delaying the healing process, increasing risks of infection throughout the body of that patient. We also have to bear in mind as well, when we're looking at these irrigating solutions, we've got to get a nice balance between the antimicrobial performance and the effects they're having on mammalian cells. And we call this a therapeutic index. And this is, of course, is what is used for drugs. And it's only slowly now getting into the arena of antimicrobials. And what we've got to make sure is that you're getting maximum therapeutic effect on killing the bugs, but at the same time having low cytotoxicity in terms of effects on cells. And what we do know when we look at therapeutic indexes and when we look at, often it can be called the biocompatibility index, but we know from reports out there that things like povidone iodine, chlorhexidine, sulfadiazines that are used in wound care, these can have quite a low therapeutic index, whereas things like PHMB have a high one, which means they're very effective at killing bugs and they have a low effect on human cells. So you're getting a good balance in terms of improving the healing process. And as I mentioned before, it's very important that we understand any cross-resistant issues of antiseptic usage and that of switching on and selecting for antibiotic-resistant genes. So there's lots of documents out there, lots of papers which can show you the major antiseptics that the FDA now are looking at, whereby if you get to sublethal concentrations of those antiseptics, they can actually switch on antibiotic resistant genes. So in conclusion, when you're looking for the right irrigating solution, it's very important we have this biofilm issue in mind, that we're trying to prevent that being formed, but at the same time trying to stop those microbes attaching. So when we look at performance, we can highlight which are the most efficacious there preventing biofilms. We can look at the ones that are less cytotoxic, and we look at the clinical considerations there on which one is providing good, solid clinical evidence that they are effective. And reducing a step in the irrigating solution is very important. So, for example, as I said, you don't have to wash out PHMB. And because it has this surfactant effect as well at lower concentrations, that's aiding the actual prevention of those bugs attaching onto hardware and risking that patient from getting an infection. Thank you very much indeed for listening, and I hope there was something there that may be useful for you.